So enzymes are the basics, the review, the stuff you should remember but probably don't.
I said the word catabolic reaction over Christmas break to one of my aunts, and they were like, oh.
Fantastic.
I can't remember what context it was used.
I hope correct.
I think it was.
Even better. So a catalyst is a substance that helps a chemical reaction to occur, aka enzymes. And enzymes are proteins, and proteins are made up of amino acid chains. This is where the review is. That should be the duh moment. Oh, also, enzymes are a large section of the CLEP test. Or like when it says, like enzymes has a percentage number by it. Whereas like coupling from last time we talked, no, there's no number by the word coupling. But enzymes, it's like a chunk of the test. And here's your one section on it. So pedal to the metal. Okay? So enzymes work basically, in the most basic and simple sense, by lowering the activation energies of chemical reactions within a cell. So remember this little graphy poo from last go around? It tells you that your reactants have, let's switch it here, have this much energy, and then as they gain energy, the purple line is if you don't have a catalyst present, they're gaining energy, gaining energy, gaining energy, gaining energy. And right at the top, what do we call this tip of this graph again?
Apex.
Good word, but not. You're not wrong. I guess technically it is an apex, but not the word I'm looking for.
I know what it is.
It's the top.
Oh. I never would have thought
The
that.
transition site. This is where your reactants transition to products. And you need to obtain this amount of energy from the lowest point to the highest point in order for that transition to happen. We call that activation energy. Once the correct amount of activation energy has been obtained, then your reactants transition to products, and we have some amount of final energy. Okay? In this particular case, your reactants have lower energy than your products, so we gave off energy. So is this exergonic? Or sorry, we took in energy. So this would be endergonic. Very good. Now, the red dashed line is showing you what happens when you have an enzyme present. Notice how your transition site is now lower than if you didn't have an enzyme present. So this is just a graph showing you of which or how activation energy decreases when you have an enzyme present. Your reactants start at the same amount of energy, your products end at the same amount of energy. The only thing that changes is how much energy is required for the transition between reactants and products to occur. So enzymes will bind to, I'm right here, to reactant molecules, and it snuggles into them in such a way as to make the chemical bonds easier to break in the reactants and then easier to reform in the products. Okay, kind of softens them up. It makes them all warm and gooey inside and safe, so they're willing to do what they're supposed to do. Enzymes do not change the reaction's free energy, and we see that because we're same at the reactant level and same in the product level. Meaning enzymes do not change whether a reaction is endergonic or exergonic. Okay? So if the reaction is exergonic without enzymes, it will still be exergonic if an enzyme is present. Okay? So, we need to learn some terminology when it comes to enzymes. The two big new words here are substrate and activation site. The substrate is the molecule on which the enzyme acts. These are your reactants in a reaction. Reactants. There may be one or more substrates. It will depend on your particular chemical reaction. The substrate is not the enzyme. The activation site is a location on an enzyme where the substrate binds or bonds. In this activation site, there will be a unique combination of amino acid residues and side chains. And remember, side chains are sometimes referred to as R groups within this activation site. So the substrate will bind to the activation site. And when you have the substrate bonded or binded to the activation site, we have the enzyme substrate complex. Each residue inside the activation site has different properties. So it could have different sizes, big, small, medium. It could be acidic, an acidic activation site. It could be basic. It could be hydrophilic or hydrophobic. It could be a positive charge in there, a negative charge, or it could be neutral. So each of these activation sites has very unique properties. And this unique combination of amino acid residues, the positions of these amino acid residues, the order or the sequence, the structures, and these properties create very specific chemical environment within that activation site. That means that each substrate has a specific enzyme. Okay, you can't trade enzymes out. Oh, I don't have the right enzyme, we'll just use this one. No, it doesn't work like that. You have to have the correct enzyme for the correct substrate. It used to be thought of as like a lock and a key. You need the correct key to unlock a lock, right? So that's kind of a way to think about it for now, I guess. So there is a specifically matched enzyme for each substrate. Like in all good rules, there is a little bit of gray area, but we're not really going to talk about that. Oh, Brett. So, enzyme activation site and substrate specificity, environmental changes. When I say environmental changes, am I talking about out there in the woods environment, or what? Where's the environment? What? Not the woods. Not the atmosphere.
The environment is like what you're in.
What you're in. So if this enzyme is in your liver cells, your environment would be your liver. Okay? So environmental changes influence an enzyme's ability to function. Conditions need in your environmental conditions need to stay within a specific enzyme's optimal range. Guess what? Each enzyme has a different optimal range of like temperatures and pH. Remember in your 10th grade science, we did the enzyme lab where you took a chunk of liver, put it in a test tube with peroxide, and it fizzed up. And then we cooled down the liver or something, or the peroxide, and to see how reactive that reaction was then. Same with pH. Your temperature and your pH, there's a window of where that enzyme works the best. If you get outside that window, it may still work, but it's just not going to work as well. So generally, increasing environmental temperature will generally increase your reaction rates, whether you have an enzyme catalyzed reaction or not. Increasing or decreasing the temperature outside of an optimal range can affect the chemical bonds within the active site, making them less well suited to bind to substrates. If you get too far out of the optimal range, you can denature the protein, which is a process that changes the substance's natural properties. It pretty much alters it so much that you can't get it back. Once a protein has been denatured, you can't reverse it. So an example here is when you fry an egg, you have put a lot of heat into that environment, yes? So you have actually denatured all of the proteins in that egg. And we can't go back to a raw egg, can we? Because your proteins have been denatured. Their shapes have changed, their properties have changed because of too much heat. Denaturization can happen if you alter the pH too much as well. It's not just a temperature thing. So, here we finally get a picture of an enzyme and a substrate. So the enzyme is the big gray blob here. And for Brett, who everything's a gray blob, says, I will label it with an E for enzyme. The active sites then are the little divots in the enzyme. And then you can see we have a substrate. So, like I said, this used to be thought of as like a lock and a key. They fit, you just shoved it in and they fit nicely together. However, upon advancements in technology, what they really realized is we have something called an induced fit. This is a dynamic fit between the enzyme and its substrate, in which both components modify their structures to allow for ideal bonding. So as the enzyme and the substrate come together, they will interact that causes a mild shift in the enzyme structure. And when I was typing this, all I could think of is like when right now Grant claims he's really into snuggling. I want to snuggle you, mommy. So when he crawls up into my lap, and then he kind of does this little like wiggle thing, and I kind of just, wrap around him a little bit more. I feel like that's what the induced fit is. You just kind of just resituate just a little bit to make that fit just a little more snug.
When a cat goes like this too?
I guess I don't have experience with that, so I'm maybe not.
Do you guys know when like a cat like crawls up on you and they're just like, and then they just like sit?
Oh yeah. Their dogs do that too when they go to sit down, like they move around a little bit.
Yeah, and when they finally lay down, they kind of just give. I think if you crawl into your bed, you do it too. You lay down, but then you do that last little, just adjust. You get that pillow in there just right, and the blankets wrapped around just right. You all know what I'm talking about, but you might not admit it. So, anyways, what you should also notice about this diagram is how your substrate, this is a two-piece substrate. We got the little rounded edge and then the triangular piece. So it fits into the activation site, snuggles in tightly, which now we have the enzyme substrate complex. The reaction proceeds. And now we have the blue rounded piece and now the yellow triangle piece, indicating we have gone from the green reactants to the blue and yellow products. And once the reaction is completed, the enzyme releases the products. And now this enzyme can be used again because it looks identical as to when it started. Enzymes are reusable. They're reusable. It's not a one and done thing. So, when an enzyme binds to its substrate, it forms what we call the enzyme substrate complex, and there are 4 ways in which the enzyme will lower the reaction's activation energy. So, the first way is that the enzyme can promote chemical reactions that involve one or more substrate by bringing the substrates together in an optimal orientation. So they can just line up, make sure the correct molecules are as close to each other as possible so that they don't have to travel further. Does that kind of make sense? The second way is they can create an optimal environment within their active site. So maybe the reaction needs a little bit more of an acidic environment to happen in order for the reaction to proceed than what it's in. And so in that specific activation site, that enzyme will make just that little area a little more acidic, so then bam, that reaction can happen. The third would be contorting substrate molecules in such a way as to facilitate the bond breaking. to help reach the transition state. So it could just cause, help bend the reactant molecules just so they're a little closer to their bond breaking point. Remember how I said I think of like a glow stick every time? And so maybe it's just bending it just right before it would crack. So that way it helps reach the transition state faster. And the fourth way is that it can actually be part of the chemical reaction itself. So we've talked about how a lot of these reactions aren't just a one-step process. There's maybe two or three kind of middle reactions that have to happen before you get your end product. And so sometimes the enzyme will take part in the chemical reaction. Amino acid residues in that activation site can provide certain ions or chemical groups that actually form covalent bonds with the substrate molecules as necessary or as a necessary step of the reaction. No matter which of these four ways an enzyme can function or will function, it will always return to its original state after the reaction is done. So it will always be able to be reused. Yes, yes? The relative amounts and functioning of a variety of enzymes within a cell ultimately determine which reactions will proceed and at which rate. So if you need a reaction to happen, you need to have the enzyme available. If the enzyme's not there, that reaction isn't going to happen. So this is a very controlled process. Your environmental factors, such as your pH and temperature, will partially control enzyme activity. And then there's also other mechanisms within the cell that can facilitate whether the enzymes will function or not. So regulation can either promote or reduce enzyme activity. Competitive inhibition is a type of inhibition. If something's inhibited, does it work or not? It does not. So a competitive inhibitor is a type of inhibition in which the inhibitor competes with the substrate molecules by binding to the enzyme's active site. So that's down here. So the substrate that should go in this enzyme is this top one. So the yellow, the pointy end goes into the active site. Your product then is the diamond and then the little Pac-Man piece. However, if you have a competitive inhibitor in there, notice how we still have that diamond shape to enter into the active site. It can trick it because it fits, but it's a different substrate because we now have the square end, which if that's plugged in there, it's blocking the active site for the desired substrate. Does that make sense?
So basically it's like a cock going to the drug bus before the drug people get there.
Yes. Allosteric inhibition is inhibition by a binding event at a site different from the active site, which induces a conformational change. Remember, what does conformational mean? Shape. Remember, conformation is just a fancy word of saying the shape of a protein. And we can say that because enzymes are proteins. So, if we have allosteric inhibition, what happens is there's a site away from your active site that this inhibitor can sneak into, and when it sneaks into, it will change the shape of the active site, and now this substrate can no longer fit into it. Okay, so it's not plugging up, it's not blocking the active site like competitive inhibition is, it's just altering the shape. So this inhibition is a negative thing. However, we can have allosteric activation in which we have an activator plugs into that allosteric site, which now then makes your active site the correct shape for your substrate. So this allosteric can go either way. It can be helpful or it can be not so helpful. I guess the big thing to realize is that allosteric is away from the active site. The competitive is in the active site. Most enzymes require helper molecules in order to work properly. These helper molecules are called either cofactors or coenzymes. A cofactor is an inorganic ion that is required. Commonly, we have iron or magnesium ions, usually Fe2+ and Mg2+ are the two main ones. However, the example I'm giving you is a zinc ion. The enzyme that builds DNA molecules, called DNA polymerase, requires a zinc ion, Zn2+, to bond to it in order for it to function properly. A coenzyme is a small organic molecule which is required. Usually these are your vitamins. Example, vitamin C is a coenzyme for multiple enzymes that build collagen. Collagen is the stretchy fiber in like your skin. It makes it spongy, so to, like it can stretch as needed. Go.
Vitamin C. No, vitamin D is the sun.
Yes. Okay. Here's just a brief picture of various coenzymes that you get from like your dietary vitamins. Like if you take a supplemental vitamin or a multivitamin, you are pretty much taking these coenzymes in to help your enzymes out.
Why do they taste so
Because they're not flavored. I don't. Enzyme compartmentalization just is telling you that different enzymes are stored in different organelles based on what reactions happen in that particular organelle. So, for example, the enzymes involved in cellular respiration, which is breaking down that sugar, which happens in the mitochondria, those enzymes are going to be found in the mitochondria, compared to enzymes involved in digesting cellular debris in foreign materials, which would be found in the lysosomes, because lysosomes are the recycling centers. They break things down. So remember, since enzymes are so specific to the reaction in which they work for, they will also be specific to where those reactions happen. Make sense? Last thing is feedback inhibition. So where do the molecules for allosteric and competitive inhibition come from? Well, they can come from your prescription medication or your non-prescription medication. It can come from toxins and other poisons in the environments, but it can also come from products within your metabolic pathways. So the first three, the meds, the toxins, and the poisons, that's all just bad luck stuff usually. If there's a toxin in your body, it's probably going to be an inhibitor, a competitive inhibitor, or something that changes how fast your body reacts or the reactions are happening in your body. Sometimes products from your cellular metabolism will actually be an inhibitor to the reaction overall. This is called feedback inhibition. So, if you follow the picture, start with your substrate. Your substrate binds to your enzyme in the activation site, and then it makes your product A. And then, because remember, metabolism is a series of many steps. This now, different color, notice, we now have a secondary reaction going on. They call it an intermediate. So the product from this first reaction becomes the reactant to the second reaction, so a little domino effect. It plugs into the activation site. We do enzyme 2 does the reaction, which produces product number 2, the blue one, which then goes into enzyme number 3, does the third reaction. and we get our end product, the desired product. However, this end product can loop back or feed back into the enzyme and be an inhibitor. Is this a diagram of a competitive inhibitor or an allosteric inhibitor?
Yes.
Allosteric. Why?
Because it's not competitive. There's nothing that can take its spot.
So this is allosteric because it binded away from the active site. So the active site is right here, and it changed the shape. Instead of it being triangular now, it's square. So can this triangular substrate fit into it? No. So allosteric because it's away, and it's inhibitive because our substrate can no longer fit into the active site. The other thing that is showing you is how we have this feedback loop, how the product actually can inhibit the reaction from occurring any further. And this is a real thing in your bodies, and it's a good thing usually. So we see this when we produce amino acids and nucleotides. It's controlled through feedback inhibition. If we don't need amino acids to be produced, or a particular amino acid, the product will go back and plug up the enzymes so that it can't be used because it doesn't need to be used. But then as soon as it needs to be used, the inhibitor will be released and the process can start over again. Yes? Same thing. ATP is an allosteric regulator of some of the enzymes involved in breaking down sugars. So when it plugs in, it can stop the breakdown of sugars if we don't need to be breaking down sugars. And then ADP serves as a positive allosteric regulation for some enzymes that ATP inhibits. So they can work hand in hand. If they need to, ATP is blocking or inhibiting the production of whatever, and it's time to start making it, ADP will plug in, kicks the ATP out, and now the process can start making whatever it's supposed to be making. So that is your enzymes in a nice 27-minute video.